Hello students, welcome to my YouTube channel, and in today's lecture we shall discuss about cell division. Cell division. So what do you mean by the cell division? Parent cell giving rise to the daughter cell is called as, or we discuss those events in an heading called as cell division. So cell division, it is of two types. We can see two types of cell division in all the living organisms which reproduces sexually. That is, first cell division is mitosis and second cell division is called as meiosis. There are two types of cell division we can see in almost all the living organisms which reproduces sexually. The first one is mitosis and the second one is meiosis. Mitosis is a type of cell division that takes place only in the body cells or somatic cells. Body cells or somatic cells. The division which is taking place in my skin, the division which is taking place in my muscle fibers, all the division is called as mitotic cell division, taking place only in the body cell or somatic cell. One more is the meiosis, the division which is taking place in the reproductive cells or gametic cells. Then such type of division is called as meiosis. First, we shall go to mitosis. Mitosis, as you know, it takes place in the body cells. Mitosis is also called as equational cell division. Mitosis is also called as equational cell division. It is also called as equational cell division. Why it is called as equational cell division? You will be knowing it after the ending of the topic. Okay, first, mitosis is subdivided into two substages. First one is interphase and next one is young phase or mitotic phase. M phase or mitotic phase. Interphase, it is called as a preparatory phase. It is called as what? Preparatory phase. It is again subdivided into three substages. First one is G1 phase, S phase and G2 phase. All the things about the preparatory phase we have discussed in our cell cycle video. Please go and watch that video and you can get the link in the description. Then M phase, it is again subdivided into two substages. The first one is karyokinesis and another one is cytokinesis. Karyokinesis is again Karyokinesis is called as division of nucleus. Cytokinesis is called as division of cytoplasm. Karyokinesis is again subdivided into four substages. The first one is prophase, metaphase, anaphase and telophase. It is subdivided into four substages: prophase, metaphase, anaphase and telophase. Now we shall go deep into the karyokinesis. During prophase, what all events takes place? Nuclear membrane and the nucleolus starts to disappear. Nuclear membrane and the nucleolus starts disappear. Centrioles which have duplicated during the interphase, they start to move to their opposite poles. DNA which is present, it starts to undergo condensation. DNA which is present, it starts to undergo condensation. DNA will be an elongated structure in the normal cell. When the cell enters into the cell division, when the cell division starts, at that time the length of the DNA is reduced by undergoing condensation, and after condensation, DNA, elongated DNA, it is converting into a chromosome. Next, this is about the prophase. At the end of the prophase, nuclear membrane and nucleolus is completely disappeared. Nuclear membrane, nucleolus is completely disappeared. The centrioles, they have completely moved to their opposite poles. Centrioles have moved to their opposite poles. DNA condensation is complete. DNA condensation is complete. After this, the cell enters into the next stage called as metaphase. When the DNA condensation is complete, the DNA will be converted into chromosomes. DNA, it undergoes condensation and forms chromosomes. Centrioles which are present, they produce spindle fibers. They produce spindle fibers. These spindle fibers are attached to the kinetochore of the centromere of the chromosomes. There is, if this is a chromosome and this is a centromere, within the centromere there is a disc shaped structure which is present, that is called as a kinetochore, and the spindle fibers are attached to kinetochore of centromere of chromosome. Now, at the end of the metaphase, all the chromosomes are arranged at the equatorial plane. All the chromosomes are attached at the equatorial plane or placed at the equatorial plane with the help of these spindle fibers. They come exactly at the center of the cell. This is a distinguishing feature of metaphase. After completion of the metaphase, the cell enters into the next stage called as anaphase. Cell enters into next stage called as anaphase. During anaphase, the spindle fibers which are produced by the centromeres during the metaphase, these spindle fibers these centrioles starts to take back or withdraw the spindle fibers. They start to withdraw the spindle fibers. They starts to withdraw the spindle fibers. As a result, tension is created at the centromere and chromatids will cut. The centromere breaks and chromatids gets separated. See here, due to the tension which is created, the chromatids of the chromosomes, they get separated. Chromatid breaks and chromosomes get separated. But still, the spindle fibers are still withdrawing back by the centromere. See, if this was the complete chromosome, these two are called as chromatids. These spindle shaped structures, two spindle shaped structures are called as chromatids. This centromere breaks, upper chromatids go towards upper side and lower chromatid comes towards lower side. This is the distinguishing feature of anaphase, that is breaking of the centromere, chromatids moving to their respective poles with the help of spindle fibers. Then after this, the cell enters into the next stage called as telophase. During telophase, reversal of prophase events takes place. Reversal of prophase event takes. What all the events that took place during the telophase? Nuclear membrane, nucleolus started to disappear. Here, nuclear membrane and nucleolus starts to reappear. Nuclear membrane, nucleolus starts to reappear. Centrioles which were moved to their opposite poles, they starts to come to their original position. They starts to come to their original position. The DNA which was present in a condensed state, now it starts to undergo decondensation. It starts to undergo decondensation and nucleolus is again formed. Nucleolus is again formed. See here, the reversal of a prophase event taking place during the telophase. By the end of the telophase, karyokinesis is complete. By the end of the telophase, karyokinesis is complete. I mean, division of a nucleus is complete. Then the cell enters into the next stage called as cytokinesis. During cytokinesis, a furrow is developed exactly at the outer surface, at the center of the outer surface of the cytoplasm. This furrow deepens. And finally two daughter cells are formed. Two daughter cells which are present. Division of nucleus, a furrow deepens, and finally the daughter cells will be formed. Two daughter cells will be formed at the end of cytokinesis. So, mitosis, it started with a single parent cell. Mitosis, it started with a single parent cell, isn't it? It started with single parent cell, and now it is having two daughter cells. It gave rise to two daughter cells. So this division is called as mitotic division, or earlier of the video I told that it is also called as equational cell division. Mitosis is also called as what? Equational cell division. Why? Because the daughter cells which are produced at the end of the cytokinesis will be very similar to their parent cells. In all aspects, especially with respect to their chromosome number. Say this chromosome in this parent cell, the chromosome number is 12. Here also the chromosome number will be 12 only. There will be no change in the number of chromosome. Hence, this type of cell division, where almost all the aspects which are present in the parent cell, they are present also in the daughter cell. So, such a type of cell division is called as equational cell division. Mitosis is also called as equational cell division. This mitosis, always the parent cell which undergoes mitosis, it produces only two daughter cells. At the end of the mitosis, we will get two daughter cells. And this mitosis helps in growth of the organism. If earlier in our embryonic stage, our life started with a very small size of nearly 1 centimeter and 2 centimeter, and earlier when we were in the womb of our mother, our life started with a single cell, and now we are having trillions of cells, and those changes were taken place only because of mitosis. So the mitosis is called as equational cell division. Thank you.